olfactory bulb sits on top of that cribriform plate. It gives rise to the olfactory nerves that come down through those cribriform foramina into the nasal cavity, and it's the axons of the olfactory nerves are situated in between that nasal epithelium that line the nasal cavity.
So where are the cell bodies for those axons?
Right here, so within the epithelial
Oh,
cells,
okay.
yeah. So what kind of nerve cells would you classify these as? Unipolar, bipolar, multipolar?
Bipolar.
Right, because we have a cell body, projection one way, projection the other way. Yep, so we have bipolar cells for the olfactory nerves. So, do you understand kind of where this all fits in anatomically with where the cribriform plate is, where the olfactory tracts arise from, olfactory bulbs sitting on top of the cribriform plate? Does that all make sense?
And those are referred to as tracts because they're still like part of the brain. Like when the nerves are
Because,
not part of the
yeah, it's the
synapse, right?
initial projection off the brain.
Okay.
Yeah. Yeah.
See, the olfactory nerve endings are like in your nasal, in the olfactory.
Correct, yeah, so
region.
I would go, so you go olfactory tract arising from the brain, and you have the olfactory bulbs, which are those, enlargements, and then, I guess I don't know if you can, it's olfactory bulbs, and then olfactory nerves are the ones, yeah. So for classifying those terms. Same thing, just different picture. So there's 1,000 different types of smell receptors, and they register a signature pattern, and then your brain recognizes that pattern. So there's not a smell receptor for hot dogs, a smell receptor for whatever else. It's a combination of certain receptors firing in kind of like a code or pattern that is designated one smell or another smell. Does that make sense? Onto the eye and vision. This is going to be the largest portion. So vision is the dominant sense in humans. 70% of sensory receptors in humans are in the eye. And that's not just special senses, it's all sensors and fire. 40% of the cerebral cortex is involved in the processing of visual information. The eyeball is obviously the visual organ. It's about 1 inch. So
Why is Alex's name on it?
different Alex,
Oh.
but it just occurred to me that they were kind of similar personalities.
That's why I was laughing. I'm like, did you just add that in there last
So
minute?
somebody-
I eyeballed it,
one of my
Alex.
students in a previous semester said that, and so I typed it in there and I said, I'm gonna steal that, but he wanted credit. So I gave him credit. That is funny, he was a lot like Arnold.
And his name is Alex. I think there's just something.
You
with
should
an
There's
Alex.
something about it, yeah.
You should delete that, and when he comes in, go over this slide again and see if he says it.
I think we should. Is he coming in?
Yeah, he's coming in.
All right. We're all gonna freak out. We're all gonna pause.
We're like,
Why's everyone looking at me? Why's everyone
Alex.
eyeballing?
why is everyone eyeballing me? I need a pun in my extra credit thing at the end. What did I say? I don't know, but I'm like dot dot dot. Pun intended.
Slide it up.
I have to look at it.
You guys can't smile. We're all smiling.
Well, for people who can't see, we can smile. Like, I'm gonna smile, but I'm not gonna look at him. I'm like,
This is all gonna be on the recording too.
Right,
well,
lies
he's
in the
gonna see
bony
the recording
orbit.
afterward.
Let's keep going. Lies in the bony orbit, surrounded by a protected cushion of fat. So obviously, we don't just have the eyeball itself, we have muscles attached to it, we have nerves going to and from it. We have different structures around it, like the eyelids or the palpebrae, we have our eyebrows. So the eyebrows are just thought to kind of be protective, block light. Eyelids or the palpebrae, so lower and upper portion, and the opening between the two is called the palpebral fissure. So obviously that fissure is the opening where we see the eyeball exposed. Then eyelashes, again, protective from dust and particulate. The tarsal plate is a connective tissue structure that is embedded in the palpebrae, the eyelids, and acts as an anchor point for the muscles that operate the eyelids. So those muscles, orbicularis oculi, acts to close the eye, levator palpebrae superioris, acts to open the eye and lift that upper lid. So tarsal plate is an anchor point, connective tissue. It's in the eyelids. These muscles attached to it and use it to help open and close the eyelids, the palpebrae. Tarsal glands, these are modified sebaceous or oil glands in the tarsal plate. They help to lubricate the surfaces of the eyeball and the palpebrae to limit friction. These are not tears, so don't confuse this with tears. We'll come to that. This is an oil, not water. The conjunctiva is a transparent mucous membrane, stratified columnar epithelium. So what would that look like? What would that epithelium?
Multiple layers.
Multiple layers. Yeah, and then columnar lower than the other eye.
You said stratified?
Stratified, yeah.
Stratified, sorry.
Okay. So the conjunctiva is a single layer that folds over on itself. So you have a layer on the eyelid, then it folds and then covers the exposed portion of the eyeball, specifically the sclera, so everywhere but the cornea. So the palpebral conjunctiva covers what surface, do you think? The back of the palpebrae, the back of the eyelids, and it folds on itself. And then bulbar conjunctiva covers
The sclera?
the eye itself, that sclera on top.
I told you. I do.
Okay, the lacrimal apparatus, this is what's going to produce the tears that help clean the eyes. So fluid has mucus, antibodies, and lysozymes, so it's not merely just the water. It has other things that help keep the eye clean and assist in your immune function. So the lacrimal gland is in the orbit of the eye, and it's at the superior lateral margin of that eye area. So the tears are produced and ejected from here, so superior lateral, and then they migrate across the eye. From there, they get to the medial area of the eyeball. They pass through the puncta, which are the little openings on your medial eyelids. If you ever looked in the mirror, have you noticed those tiny two little holes? Those are the puncta. So those are openings into what's called the canaliculi, which are these initial tubes. So puncta, the opening, open up into the canaliculi. Did you guys find them?
Yeah,
Just poking yourselves.
we're just poking ourselves in the eye.
Okay, so canaliculi empty into the lacrimal sac, and then the lacrimal sac empties into the nasal cavity through the nasolacrimal duct. Okay? So your eyes are continuously doing this to keep your eyes moist and clean. And then obviously if you have, typically it's a result of an emotional response, right? You're sad or whatever. You just out.
You're sad or whatever.
You can cry because you're happy too, which I've never done in this class.
Wow.
I don't think you've
I didn't
shown
know
any
robots
emotion.
Drag us while you're at it.
I don't think you've shown any emotion other than just like.
No.
When I say something mean and then I laugh. I do that.
Well, that doesn't mean you're happy. It just means you're like a passive aggressive.
That's the only time I'm happy in my life. Is when I'm in here being mean to you.
Oh, so
Are
when
you
you're being
okay?
a bully,
Is
when you're
everything
being
okay at
when you
home?
when you enjoy taking pain out of others, that's
You
when
need
you're
to
happy
talk.
the most.
It's okay.
Yeah, let's have a
Everyone
therapy
has their
session.
issues.
Now we can analyze your pain.
Let's do it. Let's
You're hiding your
Wait,
pain.
We need a
There's none of
demonstration of the lacrimal ducts
So,
and shit.
so emotional response is the lacrimal gland just outpaces the,
Wish I had
the
time for him to
reabsorption.
heal up
Your mom
his
never
like
loved you.
childhood trauma.
He's textbook.
He's textbook.
Textbook what? It's funny because it's true.
Ooh, I'm so
Psychopath.
It could be both.
He's an
They're different-
infomaniac.
there's
He
different
just looks like
diagnoses.
I
everyone.
swear, every semester, the last class, you guys get all mouthy. Every class.
Because it's the
Every
last.
semester.
That's the last day.
Yeah.
We're
No,
just
we've
like,
been
Yeah,
out
you
okay.
take
this
your
entire
final, I can still
semester.
rake you guys over the coals. It's not over.
No,
Did you say rape?
no.
Rake.
Break,
Nah.
break.
Rake. Come on.
Alright, innervation. So what
That one was
nerves
fun.
participate in eyeball function? So movement as well as the sense of vision.
Wait,
So let's start with eye movement. What nerves, cranial nerves are involved in eye
3,
movement?
4, 6.
Oculomotor.
Oculomotor, which is 3.
Abducens.
Yeah, abducens, which is 6, and then
Trochlear is 4.
trochlear is 4. Yep, and then what's the nerve of the actual sensation of sight?
Optic.
Optic.
Yeah, number 2, optic.
You know, Henry was calling.
So 3 layers of the eyeball, and we touched this in lab, so should be familiar. So that the outermost layer, the most superficial, is called the fibrous layer. It's dense connective tissue. So we have the sclera, which is the white of the eye that covers the majority of the eyeball. And then the cornea, which is the clear central
bulge.
portion that covers the iris. So next layer, middle. So we're going 1 layer deeper into the center of the eyeball. That is the vascular layer. So it's made up of the choroid, the ciliary body, and the iris. So the choroid is just that middle vascular layer in between retina and sclera, which again covers the majority of the eye. And you get to ciliary body. What was the ciliary body? Yeah, it's a circular sphincter muscle. It does what? Yep, it attaches to the lens.
Holds it up. I don't know.
So it attaches through those suspensory ligaments
Does he look like
to
that?
the lens. If the muscle contracts, what does it do?
It doesn't die.
Tenses the ligaments, causing what of the lens?
Expansion.
Change in shape, specifically,
Narrower.
yeah, flattening. Yeah, which, yeah, if you're cross-section, it could be And then obviously if it relaxes, less tension,
If you smoke a
it
bunch
takes that
of
more
weed.
curved shape. So that changes how the light refracts through the lens
Doesn't help.
and how it's gonna focus on the retina.
Stop it.
And the iris,
Can I ask you- So when you're like in a really bright place and your pupil dilates, is that a function of the
That's-
ciliary bodies or
no.
the, no?
yeah, that's separate, so that the pupil is gonna let more or less light in, depending on the light around you, to prevent damage, and it affects visual acuity, right, if it's not functioning. But that is separate from the lens. The lens is gonna be posterior to that, and it's gonna change how the light bends, but not necessarily the amount that's coming in.
Yeah, so it's changing the shape of the lens.
Correct. Yep. Good question. So iris, pigmented area except for the pupil. So the sphincter of the pupil, so the opening of that, and the muscle that controls that is under parasympathetic control. so when it contracts, the pupil's gonna get smaller. So this is in bright light, close vision situations. And then the dilator of the pupil, the one that causes it to open, is under sympathetic control. And this is for dim light to allow more light in and what light's available in, as well as distance vision.
Obviously.
Okay, and then the inner layer is the nervous layer. So this is the retina. This is what's actually has the visual receptors that are going to feed in and create that optic nerve. It's going to actually create a sensation of vision.
So aside from the nerve, is there anything inside the globe of the retina? Is it like empty?
Like it's a basketball, and you're asking what's inside the basketball. Yeah, there's fluid and gel-like substances in there, and I won't get into that in a second. Okay, so let's go back to that lens. So the lens is a biconvex shape. So what does that mean? Yeah, so bi meaning two. Convex is
It's convex on both sides.
yeah, convex on both sides, meaning it bulges out on both sides, compared to, like, say, a red blood cell is biconcave. So if you put a red blood cell on its side and chop it, it's biconcave versus the lens, which is biconvex. Okay, so like we said, it changes shape to accommodate for different vision situations, and it's what's going to focus that light onto the retina. And depending on the shape, the light coming through is going to bend in a different way and focus on the retina, depending if you're looking at something far away, close to you. Okay. So again, depending on the shape, focuses on the retina. Obviously, if you have some disruption of that, then it's going to focus too far behind or too far forward of the retina. That's when we use lenses to help alleviate that, to change the focus on the retina, so nearsighted or farsighted. Also, too, as we age, so technically every second, every day, every year that you get older, you're not able to see as close to your face. So a 1-year-old can see something closer than a 2-year-old and a 3, 4, 5, 6. That's why when you get older, you see people,
My mother.
they don't want to put their glasses on, so they're holding it out here because their lens has become more rigid and less pliable, so it's not able to focus on closer to the face.
Oh God, no one wanted to clap with me. Shut up, Doc.
Thanks for joining us. So we're going over that eyeball. Hey, yeah, walk in and talk to them.
I'm trying to record this. Can you be quiet, Alex? Thanks.
Pay attention
Yeah,
to
we're
the slide.
recording this. Shut up. You're like.
Okay, anyways,
Focus your eyeballs on this.
It's still there, Alex. It's just like, no.
it's making it difficult.
Hey, give it to
Okay,
me.
so the eyeball. So the eyeball is a 1-inch diameter, about. It's roughly that. There's only the anterior 1/6 is visible, lies in the bony orbit, surrounded by a protective cushion of fat. I just want to emphasize the size of it. It's like about 1 inch. So I just.
Alex, do you have any comments on doing this?
Yeah, it seems to be about an inch or so.
Yeah, but how did I arrive at that? Like, just kind of.
You eyeballed it? I'm so glad. See, I don't have to be here.
There's a backstory to that.
Just hit the undo button.
Oh shit.
So the actual slide.
Yes! What?
What? So once upon a time, last semester.
Another student named Alex said the same thing.
And you had the same type
You
of
made the
personality
joke.
as you
Well, I
did.
did fail this class last semester.
Oh no. Oh no. I have it on recorder, so you don't want to watch it.
That's too funny.
I got it on video.
So,
It's a good joke. It's a quality joke.
yeah, and we thought,
But it was
what
like,
if we could get him to say it again?
and you did, so you fell right into it. You eyeballed it.
Did he go on to great things?
I don't know, I don't care about this stuff.
He really doesn't. Leave it like that, just leave it.
Alex,
I just really
next
hope-
semester, if you have an Alex, make them say it. You just see how many
I'll
times.
just stand there and
How did I get there?
How did
Wrong
I
answer.
how did I get there, Alex? You eyeballed it. That's where you got it. Alright, we had to go back just for you, so. We were so
What,
far
what
ahead.
did you think we were doing? Like, you were just like, they're being weird. Alright, so chambers of the eyeball, this is what's gonna fill that open space. So there's the posterior cavity, which is everything posterior to the lens, and then there's the anterior cavity, which is everything anterior to the lens. So the posterior cavity is filled with vitreous humor. It's jelly-like. It forms in the embryo and lasts a lifetime. The anterior cavity is called aqueous humor. It's more liquid and it's replaced continuously. So then the anterior cavity can be further divided up into the anterior and posterior chamber. So they didn't get real creative here. So make sure you understand the difference between anterior posterior cavity, the larger spaces, and then if you take that anterior cavity, you can further divide it into anterior posterior chamber. So that the anterior chamber is between the cornea and the iris. Posterior chamber is between iris and the lens. Okay, so here's a picture of it. So cross section of the eye, hollow openings, posterior cavity behind the lens, anterior cavity in front of the lens, and then we can subdivide up anterior cavity into posterior and anterior chamber. So anterior chamber is between the cornea and the iris, and then the posterior chamber is the iris and the lens. So you see how you can subdivide that here and here.
Wow.
All right, the retina has two layers. So again, the retina is the innermost layer. It's the nervous layer where light detection is going to occur. So you outer thin pigmented layer that's full of melanocytes. These are cells that prevent light scattering. Then the inner thicker neural layer. So make sure when you visualize this, you understand. When we say the outer layer, that's the more superficial layer, the one closer to the choroid layer. The inner, deeper layer, closer to the inside of the eyeball, that vitreous humor, is the neural layer. So this, like we said, layer that detects vision. 3 types of neurons. There's photoreceptors, bipolar cells, and ganglion cells. So as the light comes into the eyeball, so this is a small cross section of the wall of the eye, right? So this area here is the posterior cavity, the opening of it, right? So we're saying light comes into the eye and then hits this retinal layer. Does that make sense? So it's going to go past these 3 different neurons. It's going to go past the ganglion cells, past the bipolar cells, until it reaches the photoreceptors. And then it's going to cause stimulation, depolarization of those photoreceptors, which then are going to propagate that action potential to the bipolar cells, propagate it to the ganglion cells. And the ganglion cells have these long axons that line the surface of the retina, and depending on which area you're talking about, it's going to travel all the way down. And once you get to the optic nerve, it's a collection of all these axons as it exits out posterior. Does that make sense? So again, light comes in, it has to go past all these cells, then it stimulates photoreceptor, sends an action potential the other way out these cells, and then back to the optic nerve. Okay, so photoreceptors, there's two different types. There's rod cells and cone cells. Rod cells are more sensitive to light. So in a dim situation, a low light situation, these are going to be utilized. And they only detect black and white, and they're not very sharp. Cone cells have high acuity and bright light. So when you have high light situation, they're responsible for colored vision. And there's three subtypes: blue, red, and green. So they're named for how they look: a rod cell and a cone cell. Okay, so this is looking at the retina through the opening of the iris, through the pupil, using an ophthalmoscope. There's different structures that you can note here. So there's the macula and the fovea. The fovea is the center of the macula, this kind of darkened portion here. The fovea is where you have maximal visual acuity. You have the greatest collection of photoreceptors. You also have the optic disc. What do you think the optic disc is?
It reflects.
It's
what
where
you see.
the optic nerve is coming out. So you actually have a blind spot in the area of the optic disc, because it's where the nerve exits. It's just those collections of those axons. So when you have, so when your eyeball detects light and the world around you in that area of that optic disc, you actually don't see that. It's a blind spot. But the brain is so good at covering up and compensating for that, you don't ever notice, right? So there is an activity you can do. You can look it up on YouTube about finding your blind spot. It's just a sequence of eye movements and finger movements, and you narrow in on when your fingers leave the visual field, and you can go to one side or the other of it and see your finger, but you can find that blind spot if you're interested.
I found it.
You put your finger behind your head, that's not the blind
I
spot.
put it right here. I can't see it.
Your eyeball is just...
You know what? This class.
Also notice that the optic disc has a collection
Put
of
it behind your head.
arteries and blood vessels. They also exit the eyeball along with that optic nerve. So if we go back a few slides, so vasculature is going to exit with that optic nerve too, the axons of that optic nerve. Okay, visual fields. So you have a left and right visual field detected by your left and right eyeball, and it will go to one hemisphere or the other. So the left visual field is detected by the right, the right side of your brain. Right visual field is detected by the left side of your brain. But each eyeball is detecting part of the left and part of the right visual field. So depending on which visual field it is and which eyeball it's coming into, it will either decussate and cross over to the other side, or it will stay on the same side. So if you have the left eye picking something up in the left visual field, it's going to cross over to the right side of the brain, because that's where the left vision is detected. If you have the left visual field being detected by the right eyeball, then it will not cross over because it's already on the right side. And then the reverse is true for the left visual field. Okay? Does that make sense? It's confusing, but you understand the
So
concept.
if I'm looking at you right now, you and the skeleton are being processed in different parts at different hemispheres of my brain
Well,
because.
both eyeballs are seeing it,
Right.
right? So if you're looking at me and that, both eyeballs are seeing the skeleton and both eyeballs are seeing me, but if it's, depending on which side of the field it is, it's gonna either cross over because it's already on, because it needs to get to the opposite side, because it's processed on the opposite side, or if it's on the eyeball that's on the same side of the visual field, then it's gonna stay on that side, because it's on the same visual field. Okay, some vision disorders, just some common ones. So you can have diplopia, which is double vision. So the eyes do not look at the same point, the visual field, so you lose binocular vision. So your visual, not the visual fields, but your what's detected in the right eye and the left eye are overlapped to create a single image. That's binocular vision. So if you ever look through binoculars and you spread it wide instead of overlapping them, that's a loss of that binocular vision where you see two separate images. Misalignment or strabismus is when you have cross-eyed, so that the eyeballs are not conjugated, they're not moving in the same pattern. One is doing it, yeah, like if you go cross-eyed. So usually this is because of a weakness of one of those extrinsic eyeball muscles, or you could have some innervation issue to one of those muscles. And then the last one, lazy eye, amblyopia. So this is when one of the eyeballs is not carrying out its movement and its focusing processes compared to the other one. So what they do for that is usually patch it. So they patch or cover up the good one and then make the lazy eye work, and that usually corrects the function if it's done early enough in development.
I don't know what you would classify this as, but there's like certain people in my family, the muscle in their eyes is like, I guess in a way weak, where they have to like kind of like do like this twitching because their eyes are trying to refocus on because my mom and my brother have it, where like they're reading something to kind of do like this like little typewriter effect with their head because their eyes are trying to focus.
Reset. I would think that's part of strabismus. Sounds like misalignment, but
Because like I don't have it, but like only just certain people have it in my family.
yeah.
Because like even with like when my brother used to do track, he would have to like kind of do this with his head just because he can't
Head
focus.
correct. Yeah, I would say that sounds like misalignment. Yeah. One of my college roommates, we knew when we had to cut him off because he'd start to get a lazy eye. His eye would start to wander. So when he was sober, he was fine. But if he got really stressed out, like during finals, if he was really drunk, it'd start to go on.
Ew.
All right,
What
Joey.
did you ew.
Yeah, Joey.
Just the way he was like, oh, it went like this.
He would have to
His eyes,
close
like,
his eyes
swung
and he was
over
like
there.
living with a feet now. Like, babe, close your eyes. I don't want to look at you right now.
Yeah, like he's a
All right, last one, the ear. So like we said, two functions of the ear, hearing as well as equilibrium. So three portions of the ear: outer portion, which is the external visible part; the middle, which is the ossicles, the little bony structures that we talked about; and then the inner ear is where we have the cochlea and the semicircular canals. The labyrinth. Okay. So sound waves come into the external acoustic meatus, that external canal, and they hit the tympanic membrane, also known as, in lay terms, the eardrum. Okay. Those sound waves vibrate that tympanic membrane and cause a displacement. The tympanic membrane is attached to what? The ossicles. So in what order? Yep. In what order is it
Malleus,
going to
incus,
affect? So it's going to start with malleus, which is attached to the incus, which is attached to the stapes. So that vibration force is transmitted through those 3 bony ossicles, and then the foot of that stapes attaches to an opening to the inner ear called the oval window, which is then gonna continue that mechanical vibration and force into the inner ear. So it's going to okay, so the foot of the stapes, like we said, is at the oval window. It causes vibration through that oval window, which is gonna affect the spiral organ of Corti, which this is where the neurons and the receptors for hearing are housed. So if you look in there and look closely at it, you see this membrane that's covering the surface called the basilar membrane. Attached to that basilar membrane are little hairs that attach to the cell bodies of these nerves. So when that vibration comes in through the oval window, it causes a displacement of this basal membrane, movement of it, which then obviously changes where the hairs are and moves the hairs, and that causes a depolarization, causes an action potential to be fired that goes into the nerve. Does that make sense? Kind of, yeah.
Does it make sense, Alex?
So let's just go through it again. So we have a sound wave coming through. It goes through where? External auditory canal, meatus. Yeah. Okay, and it hits what? Tympanic membrane. Okay? It's going to cause that tympanic membrane to vibrate, which is attached to auditory ossicles. So in order, it's malleus to the incus to the stapes.
Oval window.
Yeah, the foot of the stapes is attached to that oval window into the inner ear. So that causes a vibration into the inner ear, which does what within the inner ear?
Because there's like fluid in hairs. I feel like I've heard about that before. So there's a membrane that's on top of the hairs?
Correct. Yeah. So think of it, if all you guys stood up and put your arms up and we draped a big blanket over the top of you, and you held on to that blanket, and I came over and tugged on that blanket, it would cause all of you to move your arms, right? So that's the hairs moving, which is going to cause actually the voltage gated channels to mechanically open, and you initiate the action potential in those cells, and then send it down, sending a signal to the brain.
But then aren't the hairs like, something about like, hairs like different frequencies or like frequencies detected at a different length along the, I feel like, because I remember something hearing once about like, yeah, high frequency hearing loss or something that kills all those hair cells in a certain part. I don't know, maybe that's.
I'm not sure that's possible.
Okay.
I don't know, like pathology.
Let's test it out.
Okay, and obviously, what nerve is it going to stimulate? Sorry, I keep skipping you. What nerve is it going to stimulate? Vestibular cochlear nerve, specifically the cochlear branch. Yeah. Sorry, what was your question?
So through the
Tympanic.
membrane, through like the incus, that's all vibration.
Yep. Yeah, mechanical force. Yep.
There's like no nerves, it's literally just.
Not until you get to here.
Okay.
And I mean, obviously, your ear has some sensory receptors and some nociceptors, right? You jam your finger in your ear, it doesn't feel good, it hurts, right? So there's that kind of neurological stimulation, but the act of hearing and the neurologic reception of that doesn't start until you get to the inner ear.
So what is it that actually causes the action potential? Like, we're thinking of action potentials as like electric signals. but
Yeah,
is
so
it
you
movement?
actually mechanically, so you pull on and deform the shape of that cell surface, and that causes those voltage-gated sodium channels to open, and then sodium rushes in, and then stored.
So that tectorial membrane, that's continuous throughout all of the cochlea?
Sorry, what's your question?
So-
The basilar membrane?
Oh, so okay, because it had it labeled as tectorial, so that's. the
Oh,
same
yeah,
thing.
yeah, sorry, AKA, yeah.
AKA, so the
Yep.
but the basilar, that's continuous throughout all, it's one continuous sheet throughout all the cochlea?
No, it's within these, so within the cochlea, within those spirals, you have the spiral organ of Corti. take a cross section of that, and then you look at the lining of this organ of Cordy, that's where you have all these structures.
Oh, but the spiral itself is.
Right, goes around in that snail shape of the cochlea. Alright, so to the vestibular side of things, so still gonna go through the vestibulocochlear nerve, but obviously the vestibular branch, not the cochlear branch. So within the inner ear, we have what's called the vestibule. The vestibule is that hollow part that's next to the cochlea. Within the vestibule, you have the utricle and the saccule. These are the areas that are going to contain these receptors for certain types of head movement. And then if you get deeper into the semicircular canals, they're going to detect a different type of movement. So the macula, which is the center of the utricle and the saccule. So you have the macula, the utricle, the macula, the saccule. This is where these sensory receptors are. So static equilibrium, so just as you stand here, as either you're standing or sitting, and you're not moving, that's static equilibrium, that's detected by these areas. And then linear acceleration, so movement in one line is also going to be detected in this area. And then if you get into and I'll come back to this slide, but if you get into the semicircular canals, then that's going to be rotational acceleration. So if you're tilting forward and back, that's one of the semicircular canals oriented in that plane, or you're going rotationally side to side, that's another semicircular canal oriented in that plane, or if you have rotational forces in another plane, semicircular canals oriented that way. But again, so static, standing here, we're just moving in a straight line. We're going to be talking about the area of the utricle and the saccule. So the way it's detected is, again, we have hair-like nerve fibers that are sticking up into the inner area of the utricle and the saccule. And then you have what's called the otolithic membrane, including otoliths, these little stone-like structures that cover those hair fibers. So as we have movement, the hollow area of the vestibule and the semicircular canals are filled with a fluid, an endolymph, and when that fluid moves because of displacement of yourself, that will shift that otolithic membrane, again, shifting the hair fibers of those nerve cells and then causing that action potential, okay? So kind of similar to the cochlea, except it's not the vibration of sound, it's your movement in space. Or if we're talking static equilibrium, it's your lack of movement in space. So somebody with, say, like vertigo, something like that, sometimes that's an issue in this area where you have otolithic movement when you're not supposed to. So it makes you feel like the room is moving or you're moving when you're actually not. And then the rest of your system detects, so visually and other proprioceptive receptors in your body, they detect you're not actually moving, but the otolithic issue, like the little stones are moving around and causing a faulty signal telling your system that it thinks it is moving.
When you're dizzy, is it like the fluid doesn't stop?
Again, so it depends the cause. There's different causes. So the most common cause of vertigo, where it feels like the room is spinning, is because you have some otolithic disruption. Now, if you're talking like you're intoxicated or something like that, that's dysfunction at the neurologic level. So either the pathway or up in the brain, you poisoned it and it's not detected properly. That's what getting drunk is, or any other sort
Ooh.
of drug, is you're poisoning yourself, right?
Yep.
I love it. What?
I'm not being critical. It's a good thing.
It's a good poison.
Alright, and then semicircular canals, going back to these. So instead of linear acceleration or static equilibrium, semicircular canals detect rotation. So it's similar. These are filled with that fluid, that endolymph, and you have what's called the cupula, which is an extension of these hair-like cells again. That is displaced by the movement of the endolymph, which causes the hair cells to move, mechanically open the old channels, and then the action potential from there.
So there's no otoliths in the semicircular?
Now, otolith is contained within that vestibule area. Yeah.
Within the macula?
Yeah. Okay. And obviously that action potential is fed back to the vestibular nerve, cranial nerve, back to the root. We did it. Slip clap, is that what you're doing?
Yeah.
Any questions?
Did you start over? I missed
Fucking stupid.
So in terms of the anatomy for the exam, do you know, like,
I'm sick.
all these things, like,
Dumbass.
charts, or just the ones that are sort of bold?
Let's go. The ones that are written out, like that I've written out. So some of these diagrams may have more detail potentially. It's what I've typed out that I want to. Anything else?
Since we're reviewing on Thursday, got to do some practice questions for all.
Yeah, so Thursday we'll do some practice questions for the new material, and then I'll get that review outline posted. So come with questions for either new material or stuff that you want to go over.